Dear students, in the previous module you have become acquainted with what is forensic chemistry and some historical aspect of forensic chemistry. You have become familiar that what are the main roles of a forensic chemist and then the scope and significance of forensic chemistry. And we have seen an overview of the different techniques that are employed in forensic chemistry and some of the cases that are generally encountered in forensic chemistry. So, let us continue this study with this module, and if you look at the learning outcomes of this module, after studying this module you shall be able to know how to collect and preserve the samples for forensic analysis. You shall be able to know how to maintain the integrity of these samples, what is the theory behind the different forensic analysis, and what are the different instrumental techniques which are employed in forensic chemistry. So, let us begin with that how the sample collection can be done. Evidence collected from the crime scene can be so varied in shape, size, type, delicacy and the degree of perishability that there can be no set procedure laid down to cover all the aspects of sample collection. However, all evidences should be collected keeping in mind some of the very important points, that is, crime scenes should be preserved as soon as the forensic scientists arrive to avoid the unnecessary manipulation of the site. This is very important. And whenever you are handling any article, then gloves should be used, otherwise your fingerprints may be imparted on the other articles, or if your hands are dirty they may spoil the evidence and so on. Every article should be handled as little as possible to maintain the integrity of the crime scene evidence. Careless destruction of every article should be prevented. Each article should be packed separately and labeled accordingly so that the individuality and identity are maintained. When we talk about the packaging should be secure and nothing should penetrate through it to contaminate the contents. The containers used should be strong enough to avoid damage in the transit. The top side of the box containing these containers should be always indicated and marked handle with care, and if possible you say that this is the top part, this is the bottom part, so that there may be some evidence if you put them upside down they may be damaged. So, let us study more about the handling of these evidences, that is their collection, how further, what is the theory behind the forensic analysis, and what are the different instrumental techniques which are employed in forensic chemistry with the help of graphics and visuals.
Moving on to sample integrity. Forensic chemists, in order to reduce the risk of contamination of sample, perform their analytical work in sterile conditions. In order to control the changes of tampering, forensic chemists must maintain the chain of custody for each sample. A chain of custody is a document that maintains the record of all evidences from the point of its discovery at the crime scene to its recording and transporting to the lab for examination and its temporary storage till it is finally submitted to the court. Along with other information, it also contains identification details of all the people involved in transport, storage, and analysis of the evidence along with their signatures, time, and date. This rules out the possibility of manipulation. It not only enhances the dependability of a forensic chemist's work, but also strengthens the evidence during trial. Now, moving on to theory of forensic analysis. After evidences are collected from the crime scene, these are sent to the forensic lab for analysis. The forensic chemist follows a specific process based on the scientific method for analyzing evidence. Samples collected from a crime scene and brought to the lab for analysis are called question samples because the identities and origins of those samples are unknown. In order to draw conclusions about the identity or origins of question samples, the forensic chemist need known samples as references, which are known as control samples. A control sample might be collected as part of the evidence. For instance, a hair sample collected from a suspect. Forensic analysis may be performed to identify a question sample or compare a sample under question to a known sample for the purpose of determining the source of its origin, that is, where it came from. The results of such comparisons can link a question sample and a several known sample either to a class of samples with several possible origins or to a single origin, that is, individualization. Thus, a forensic chemist has to analyze much more than the question sample. A forensic analysis follows the order, identification, classification, and individualization. The challenges found during each phase of analysis are different for different evidence. Often, identification is straightforward and obvious to the untrained eye. For instance, hair, soil, various similar looking drugs. But the fact is that expertise and sophisticated instrumentation are required for making a proper and accurate analysis. Let us learn about the importance of identification in forensic analysis. When a question sample is submitted to crime laboratory for analysis, the first task is identification. For example, if a white powder is submitted for analysis, the primary objective will be to determine its identity. If the powder is suspected of being a controlled substance, the forensic scientist will carry out a series of analysis to identify the powder. Since each drug has a different set of physical and chemical properties, an entirely different series of analysis is required to identify each drug. The forensic scientist must first make an educated guess as to the identity of the substance. Two types of analysis can be used to identify the substance: presumptive and confirmatory. Presumptive analysis. Presumptive analysis looks at the chemical and physical properties that are not unique enough by themselves for identification, but that provide enough information to narrow the search. For example, If the powder reacts with sodium nitroprusside in the presence of sodium bicarbonate and produce a very deep blue colored product, the forensic scientist may guess that the question sample might be methamphetamine. However, a number of similar compounds, all containing a nitrogen atom with one hydrogen atom and two attached carbon atoms, will also produce a deep blue colored product. This analysis does not confirm that the substance is methamphetamine, but it certainly narrows down the number of possibilities. Now, the forensic scientist can proceed to more conclusive tests, knowing that he is on the right track. Presumptive analyses are usually quick and inexpensive to perform. Another example of a simple and popular presumptive test commonly used in forensic science laboratories is luminol test. Luminol test, a method frequently used in forensic chemistry, is that employing luminol, a derivative of phthalic acid, which detects traces of blood by reacting with metal cations. The test reagent is carefully sprayed in places where traces of blood are highly probable to be found, but it is done when all the other evidences have been collected, as it is a destructive method. Thus, a blue luminescence is observed when luminol reacts with heme group of hemoglobin. However, luminol, due to its inherent quality, may react with other metal ions to give false positive results, and that is why it is only used as a presumptive test for the analysis of blood. 3-aminophthalate anion is the final product of this reaction, which is in an excited state. Upon returning to the ground state, it releases energy in the form of light, resulting in blue luminescence. Now, moving on to confirmatory analysis. When presumptive analysis is negative, they exclude potential drug candidates. When they are positive, they direct the forensic scientist towards viable confirmatory analysis. Whereas presumptive analysis only narrows the possible identities of a substance. Confirmatory analysis identify a question sample absolutely. They are required for court and must be performed to convict someone for possession of an illegal substance. These analyses use those chemical or physical properties which are unique to the substance for the purpose of identification. Typically, confirmatory analysis require more time and expense than presumptive analysis. These analyses often require the use of sophisticated chemical instrumentation to elucidate the unique properties that lead to definite identification. Comparative analysis, classification, and individualization. Many forensic analyses end with identification, for example, identifying an unknown substance as a drug, explosive, or accelerant used in arson, but some proceed on to comparison. The process of a comparative analysis is to link a question sample and a control sample to a common origin. Now, moving on to class characteristics. Class characteristics are the properties of a substance that are shared by a group of substances but are not unique to all. They allow for the placing of a question sample into a class or group of several possible origins. These properties are analogous to those used when conducting a presumptive test for the purpose of identification. Not only are class characteristics common to other substances, but they may also vary within a substance. A class characteristic that varies within a substance is called natural variation. When attempting to determine the origin of a question sample, the forensic chemist must know all the possible variations of the class characteristics from a known sample. Virtually all physical evidence has class characteristics. These characteristics are more common than individual characteristics. Many items of evidence like hair, fiber, glass, soil and paint have only class characteristics. In other words, classifications are more common than individualizations. This does not, however, suggest that comparative analysis of items only containing class characteristics are unimportant. The ability to exclude is a very powerful aspect of class characteristics. If the class characteristics of many questioned items of evidence are similar to those of many samples of known origins, each additional link, even if tentative, further incriminates the suspect. Now, moving on to individual characteristics. Individual characteristics, on the other hand, are the properties of a substance that are precisely unique to that substance and could identify and individualize that substance with an extremely high degree of probability. These properties are analogous to those used when conducting a confirmatory test for the purpose of identification. Next, moving on to instrumental techniques employed in forensic chemistry. There are a number of techniques that are commonly employed in forensic chemistry. Destructive methods take a sample from object under analysis and alter its constitution. Most of the spectroscopic techniques fall into this category. Non-destructive techniques, on the other hand, maintain the integrity of the object and thus are the method of choice by forensic examiners. For example, optical microscopy and microspectroscopy are the examples of non-destructive techniques. First is nuclear magnetic resonance spectrophotometry. The basic principle of NMR is that nuclei of certain molecules absorb radiation in the region of radio frequency in strong magnetic fields. In this way, the identification of traces and even impure samples is made on the basic principle that nuclei of some molecules absorb radiation of a definite frequency. Next is neutron activation analysis. In NAA, a beam of neutrons from a nuclear reactor is directed to a sample of test material. The material becomes temporarily radioactive, emitting gamma rays that are characteristic of the composition. Analysis of the gamma radiation provides a highly accurate and reproducible determination of the content of the sample. The determination of arsenic in the hair of dead bodies that have remained buried for hundreds of years are made possible by this technique. Now, moving on to gas chromatography, mass spectroscopy. Another useful technique that is employed in simultaneous separation and qualitative as well as quantitative analysis of one or more components of an unknown sample is GCMS. A GCMS are actually two instruments that are physically clubbed together in a single unit, the so-called tandem or hyphenated techniques. In gas chromatograph or GC, a bent or coiled, packed or coated glass column is held in a temperature controlled oven of 150 to 350 degrees Celsius. A small volume of the sample to be analyzed is dissolved in an organic solvent like methanol or chloroform and is injected into the column. The high temperature of the column evaporates the volatile components in the sample, which are finally eluted out of the column along with the carrier gas. As different components of the mixture have different affinities towards the stationary phase, each substance elutes out at different, which is known as the retention time. The retention time of such eluted components can then be compared with the known standards by keeping the experimental conditions constant. In general, the GC portion of the technique is used as a tool for separation and quantitation and not identification. The next component of this coupling, that is, mass spectrometer, is used to provide the qualitative analysis of the sample components. For this purpose, the eluent of the column is fed into a mass spectrometer or MS. This highly complex instrument uses one or more methods like bombardment with electrons, heat, electrical force, light, etc. to disintegrate molecules into ions. These ions are separated in the ratio of their mass with the help of a quadruple mass analyzer and detected by an electron multiplier. This provides a distinctive pattern of fragmentation which functions as fingerprint for each compound. For the purpose of identification, the patterns so obtained are then compared to a reference sample. Now, moving on to Fourier transform infrared spectrophotometry, another instrumental technique that assists in identification of compounds is the FTIR. In this technique, infrared radiation is used to bombard the sample. Polar bonds present in organic compounds have a natural vibrational frequency that is similar to the frequency of infrared radiation. When both these frequencies match, the amplitude of the vibration increases and the infrared radiation gets absorbed. The chart obtained as output of an infrared spectrophotometer shows the amount of light absorbed versus the wavelength. Because the intensity as well as frequency of absorption is dependent on the type of bond, the functional groups present in the compound can be determined by examining the infrared spectrum. As with the GCMS, the FTIR spectrum can be compared to that of a known sample, thus providing a key as to the identity of the compound. Next, we move on to UV visible NIR spectrophotometry. UV visible NIR spectroscopy is used to test for certain drugs of abuse. UV visible NIR microspectrophotometers are instruments able to measure the spectra of microscopic samples. It can be used to compare question samples of the evidences with the known ones. They are also employed in analyzing the papers and inks of questioned documents and to determine the characteristics of microscopic glass fragments. As these samples are not altered, UV visible NIR microspectroscopy is considered a non-destructive technique. Next, we move on to microscopy. A microscope is an optical instrument that uses lens or a combination of lenses to magnify the finer details of an object, which is otherwise invisible to the naked eyes. Microscopy is the science of revealing small objects using such an instrument. There are many different types of microscopes. Out of all, the most common is the optical microscope, which uses a light source to image the sample. Fluorescence microscope and phase contrast microscope are the brilliant examples of optical microscope. Other major types of microscopes are the electron microscope, which uses a fine electron beam to magnify the object.
So dear students, let us summarize what we have learnt in this module. We have learned that each article should be packed separately and labeled accordingly so that the individuality and identity are maintained when you are collecting the evidence from the crime scene and sending it to the laboratory for further analysis and investigation. A chain of custody is a document that maintains the record of every evidence from the point of its discovery at the crime scene to its recording and transporting to the lab for examination, its temporary storage till it is finally submitted to the court. Samples collected from a crime scene and brought to the lab for analysis are called as questioned samples because the identities and origins of those samples are unknown, while the ones which are used as reference, that is the reference samples, are known as control samples. Forensic analysis may be performed to identify a questioned sample or to compare a questioned and a known sample for the purpose of determining the source of its origin. When a questioned sample is submitted to a crime laboratory for analysis, the first task is its identification. And two types of analysis can be used to identify the substance that we have studied in this module, that is the presumptive and confirmatory. The purpose of a comparative analysis is to link a question sample and a control sample to a common origin. Class characteristics are the properties of a substance that are shared by a group of substances, but are not unique to all. On the other hand, individual characteristics are used to individualize that substance with an extremely high degree of probability. We also studied that the basic principle of NMR is that the nuclei of certain molecules, they absorb radiation in the region of radio frequency in strong magnetic fields, and in this way the identification of traces and even impure sample is made on the basic principle that nuclei in certain molecules absorb radiation of particular frequency, and one can very easily compare the NMR of the sample collected from the crime scene with the reference sample or the control sample's NMR and just compare. If it matches, it is the same substance, otherwise not. The determination of arsenic in the hair of dead bodies buried for hundreds of years are made possible by neutron activation analysis technique. Another useful technique that you have become familiar is employed in simultaneous separation and qualitative as well as quantitative analysis of one or more components of an unknown sample is the GCMS. A GCMS are actually two instruments that are physically clubbed together in a single unit, the so called tandem and hyphenated techniques. Another instrumental technique that assists in identification of compounds is the FTIR. In this, the infrared radiation is used to bombard the sample, and you can get the FTIR spectra which can be compared with the controlled sample. UVVisibleNIR spectroscopy is also used to test for certain drugs of abuse, and you have been acquainted that a microscope is an optical instrument that uses lens and combination of lenses to magnify the finer details of an object which is otherwise invisible to the naked eyes. So, once you get these evidence from the crime scene, then different techniques are employed on different types of evidence, and all these details we will be studying in further modules. Thank you.